Hello, everyone. Welcome to join PEAKS webinar today. Previously, my colleagues have introduced qualitative and quantitative proteome analysis in PEAKS. In addition, studies of post-translational modifications, or PTMs, via mass spectrometry also constitutes a wide field of research and applications. Today, I will focus on PEAKS PTM analysis by liquid chromatography coupled to tandem mass spectrometry approaches. Compared to complete proteome analysis, the major goals and challenges are quite different for PTM studies. The primary task is to identify modified peptides and proteins in a purified sample or complex mixture. Each peptide representing a modification site of interest needs to stand on its own. Since modified peptides can be of lower abundance, it is more difficult to identify from their fragmentation spectra than non-modified forms. To overcome this problem, certain enrichment steps can be added to enrich for modified peptides or proteins. Secondly, the tandem mass spectrum must contain sufficient fragmentation information to localize the modified amino acids in a sequence. Various fragmentation methods can be employed to generate such informative spectra. PEAKS support different types of fragmentation methods, such as CID, HCD, ETD, ETHCD, and etc. Besides identification, researchers often pursue to measure and compare relative abundance of modification site of interest across different biological samples or conditions. This can be achieved via label-based or label-free quantification methods, which is supported in PEAKS Q module. Lastly, it is also very useful to determine the site occupancy or stoichiometry. That means the proportion of proteins that are modified. As a data analysis tool, PTM functions in PEAKS tries to address these challenges and help your PTM studies going further. So keeping these in mind of the topics that I will cover in my talk today. First, I will show you some common practice for PTM identifications and modification site determination with PEAKS database search. Secondly, I'm going to introduce PEAKS PTM function that is specifically designed to discover more unspecified or hidden modifications by integrating PEAKS database searching and de novo sequencing. Thirdly, I will demonstrate quantification of PTMs by using PEAKS PTM profile. Lastly, I will touch a little bit on some new features that's going to be integrated in the new version of PEAKS, which we hope to make your study easier. Okay, let's start with the first topic, using PEAKS database search to identify post-translational modifications. Specifically, I will use 3 examples to demonstrate how to perform PTM analysis in PEAKS database search, with an emphasis on how the software addresses the 2 main challenges in PTM analysis, which are accuracy and sensitivity. Let's start with a simple case, modification analysis of a purified monoclonal antibody protein sample. For this experiment, we used hydrogen peroxide to treat a monoclonal antibody sample and analyzed digested peptides by using LC-MS-MS. The purpose of the study was to determine the sensitivity of the MAB protein to peroxide. We uploaded the data of control and peroxide-treated sample into PEAKS and selected trypsin as the enzyme and instrument and fragment type that were used in this experiment. In database search siting window, we input precursor and fragment ion tolerance based on the instrument that we used, and added methionine oxidation as an additional variable modification, since we knew that hydrogen peroxide could oxidize methionine residues. and then we click Finish button to let DB search run. This is the identified protein results. In this protein coverage view, supporting peptides represented by these blue bars are mapped to the sequence of the selected protein. You can click the peptide, and then its MS2 spectrum will pop up, where you can easily examine the annotated raw data. Here is a peptide containing a confident methionine oxidation modification. The M in this peptide sequence is lowercase, meaning that there is modification happened to this amino acid. There is also another unmodified counterpart identified. Here I put two spectra of modified and unmodified peptides together so that you can easily see the mass difference of Y3 and Y4 ions between the modified versus unmodified peptide spectra. The second case I'm going to show you is a bit more complex. Here I used a published dataset. Phosphopeptides were enriched by using titanium IMAC beads from a tryptic human cell line digest. Protein phosphorylation plays important roles in cell signaling systems and is a widely studied research area. Phosphopeptides, like other modified peptides, can be of lower abundance, as I mentioned before. Therefore, the author used one of the common enrichment methods to eliminate unmodified peptides in the sample. In addition, they tested a novel fragmentation scheme combining electron transfer and higher energy collision dissociation, termed ETHCD fragment method, and found that ETHCD generated richer and more confidently identified spectra. The reference paper information is provided below. So now we have added ETHCD fragmentation method in PEAKS, which will be available to users in the next version. In the database search setting, we added serine, threonine, and tyrosine phosphorylation as variable modification, and we used SwissPro human database. After the DB search is done, here is a peptide result view. So the selected peptide contains two phosphorylated serine identified by this ETHCD tandem mass spectrum. BY and CZ ion series generated by ETHCD helps to determine the phosphorylation site location, as you can see from the tandem mass spectrum and the ion match table below. Since there was a phosphopeptide enrichment step, unmodified peptides were largely washed away and were not identified for this sequence. Although enrichment step can facilitate the identification of modified peptides, there are always cases where enrichment method is not available yet, and low-abundant peptides bearing PTMs are present in a complex peptide background. So how does PEAKS perform under those difficult conditions? Can we still identify these PTMs with high sensitivity? In year 2012, we participated in a study hosted by IPRG of ABRF, which aimed to assess various software's ability to handle these situations. So they used a sample comprised of a mixture of synthetic peptides containing known PTMs, spec'd into a yeast wholesale lysate as background. And this dataset is the third example that I will show you. The two major challenges of analyzing this dataset were first, sensitivity, which means that without enrichment strategies, samples are dominated by unmodified peptides. Can softwares still identify modified peptides of lower abundance? Secondly, if there are multiple sites within a given peptide sequence that could bear a particular modification type, then can softwares correctly identify fragment ions to localize the exact site of modification? PEAKS demonstrated superior performance in terms of both the total number of correctly identified peptides and the number of correctly identified modified peptides that were spec'd in. Now, of course, we can do better than this old version of PEAKS. Here is an example showing a low abundant peptide with MS1 peak area of roughly 2E5. It has 3 potential phosphorylation sites in total. PEAKS successfully identified fragment ions that supported the phosphorylation on serine at position 3 and tyrosine at position 7 in this peptide sequence. Additionally, there were different combinations of PTMs occurred on this peptide sequence. PEAKS also identified other forms and localized the exact amino acids where PTM happened correctly. So speaking of the localization of modification site, which is another challenge in PTM analysis, I will briefly explain how PEAKS addresses this issue on my next slide. The precise site of modification can be determined by the presence of site-determining fragment ions. In this example, the presence of the B11 and B12 ions in the tandem mass spectrum determines the deamidation happens on asparagine 12. And PEAKS provides 2 options for users to determine confident modification sites. One is to use minimum ion intensity, which requires that the relative intensity of the fragment ions before and after the modification site in a tandem mass spectrum must be higher than the number you put here to call a confident PTM site. The other option is called A-score, which calculates an ambiguity score as -10 times log 10 of a p-value, and the p-value indicates the likelihood that the peptide is matched by chance. Therefore, the higher A-score, the better. So both methods provide measures of the confidence that can be placed onto the site localization. If the threshold of the method you select is met, then the PTM will be in a colored box annotated above the residue. To summarize, in section 1, I explained how PTM analysis is performed with PEAKS database search. However, the number of modifications specified by users in a database search is often limited. Otherwise, it takes too long time for the algorithm to try all possible modifications for each peptide in the database, and the number of false positive hits dramatically increase. To solve this problem, we designed PEAKS PTM function. PEAKS PTM function uses results derived from the powerful de novo sequencing algorithm and database searching. So this is a typical data analysis workflow in PEAKS. First, de novo sequencing is carried out for each spectrum. Secondly, PEAKS database or DB searching is used to identify proteins. A few highly frequent PTMs can be specified at this step. Next, the spectra with high confident de novo scores but are not assigned by database searching are mapped against the identified proteins to find additional PTMs. At this step, you can specify as many PTMs as you want. This multi-round search approach can help maximize the identification and sensitivity of your PTM studies. To enable this function, you can either check this Find Unspecified PTMs and Common Mutations with PEAKS PTM box in the database search setting window before you start the data analysis. Or if you already have a DB searching result, you can select the DB node and then click this PEAKS PTM button on the top panel. A PEAKS PTM window will pop up then you can select either all built-in modifications, which are naturally occurring modifications found in Unimode database, or choose all possible PTMs you are interested. As I said before, at this stage, the number of PTMs is not limited. You can select or input as many as you want. If you still remember, at the beginning of my introduction on PEAKS database search, I used an example of methionine oxidation analysis of a monoclonal antibody. So here we turned on PTM searching to reanalyze the data by using over 400 built-in modifications. And we found that another modification, diethylmethyl, occurred to that methionine at position 252 in addition to oxidation. Furthermore, if you zoom out to look at the data, you will see many more modifications identified by the PTM search compared to the database search, as listed in the legend and color boxed in the protein coverage view results. This is because PEAKS PTM function is specifically designed to discover unspecified or hidden modifications and maximize PTM identifications. Knowing the type of modification is the first step. You may also want to measure the modified site abundance and compare the quantity between different samples and conditions. For this purpose, PEAKS PTM profile can help to provide you with a direct visualization and summary of the quantitative information of the PTM sites identified in your studies. And two examples will be used. You can find this PTM profile button on top of the protein coverage window. By clicking this button, a PTM profile window will pop up, showing the percentage of summed, modified, and unmodified peptide feature abundance, the sequence of which contain the specific modification sites as listed on the x-axis. You can choose which type of PTM you wanted to see. Here, this figure shows the methionine oxidation quantity in hydrogen peroxide treated MAB sample, and two methionine modification sites were identified and quantified, position 252 and 428, the example that I used before. You can also choose to display two samples in the bar graph. So now we can see that methionine at position 252 is more sensitive to hydrogen peroxide oxidation compared to methionine at position 428. By turning on PTM search, we found more PTMs in this dataset. As for methionine at position 252, we identified diethylmethyl modification in addition to oxidation, which I have shown you before. So if you click the PTM profile function again, you will see the quantitative result a little bit different. Now these two bars do not reach 100% because of the identification of diethylmethyl modified forms. You can always export PTM profiling results into a CSV file by clicking the Save to Text Format button on PTM profile window. You can get the detailed information of a specific type of PTM for all proteins if you check Export All Proteins box here. Furthermore, if you check Export All Fractions box, you will get quantification result in all samples, although only one or two samples are displayed in the bar graph. Below, I pasted the CSV table exported from the oxidation profiling of MAB with PEAKS database searching and the diethylmethyl profiling with PEAKS PTM searching results. You have all the information you need for further analysis in these tables. For example, the summed, modified, and unmodified feature area in each sample, the protein accession, peptide sequence, and PTM sites. Lastly, I would like to briefly show you some new features related to PTM analysis that will be integrated in the new version of PEAKS. We hope that these changes will help you to make your analysis more easier in the future. So the biggest change is the result presentation view in PTM profile function. In the current version, you can only compare PTMs in 2 samples. However, in the next version, it will provide you with more comprehensive and detailed information and allow you to examine the whole data set. So first, on top left corner, you need to select a specific type of modification, and there will be a PTM profile table showing modified sites, summed intensities of peptide features containing the specific modification in each sample, and summed intensities of unmodified peptides containing the specific PTM site, and the best peptide score and A score. You can select to use either all peptides or only fully digested peptides from here. Secondly, in the bar graph below, you can directly visualize the results shown in the table. So the y-axis will show modification sites, followed by the sample names, and the x-axis shows modified and unmodified form abundance. It will not be limited to 2 sample comparison only, although I do have only 2 samples in this case. Thirdly, on the right-hand side, the spectra with the best PSM score from modified and unmodified peptides containing the PTM site are shown, allowing you to check the raw data easily. Surely, you can export the result to a CSV file, which contains all the information listed in the PTM profile table, such as protein information, PTM type and position, the best peptide spectral match score, and A score, modified and unmodified area in each sample, as well as some more information, for example, the protein sequence containing the PTM site, the number of and the best A scores of modified peptides in each sample. So furthermore, you can export PTM profile for all proteins by checking this box. This is what is new for PTM profile in PEAKS-DB result. Currently, in PEAKS-Q result, we do not have this PTM profile function. You may have noticed that this button is gray. However, recently, labeling-based methods such as TMT and SELEC have become more and more popular for PTM studies. In the next version of PEAKS, this function will be added to precursor ion quant, reporter ion quant for analyzing SELEC, TMT, and iTrack types of data, as well as label-free quant. Here, I'm using a TMT 10-plex data as an example first. In the PTM profile result from PEAKS-Q, the table will show you the quantity information in each group. So for this experiment, I had 2 repeats of my spec runs. Each contained 10 samples labeled by TMT 10-plex. And in the experimental setting, I specified each labeling channel from the 2 replicate runs as a group. Therefore, there are 10 groups in total, and the average value is calculated and shown in the table. Below in the heat map, you can have a direct visualization of how the modified form abundance varies between different groups. Each row is a PTM site. And the reason why we use modified forms only to generate the heat map is because in PTM studies, researchers often try to find if the modification abundance changes between different biological samples or conditions. They care less about the unmodified ones. On the right-hand side, only the modified peptide with the best peptide spectrum match score is displayed. For this case, the tyrosine at position 2 is phosphorylated, and the alanine at N-terminal is labeled by the TMT reagent. Below, all peptides containing the selected PTM sites are listed in the peptide table. For example, here, the first peptide is a fully digested peptide, whereas the second one contains a miscleavage. For each selected peptide, all features matched to it are displayed in the all matches table. Here, ions of different charges and from different mass spec runs are displayed separately. And you can see the MS1 peak of each feature in the extracted ion chromatogram figure next to it. In the export CSV file, more detailed information is provided in addition to what is shown in this PTM profile table. Here is another example showing select type of data. In this experiment, I have 3 samples metabolically labeled with light, medium, and heavy select medium, and I specified each labeling state as a group. The PTM profile result view is quite similar to what I have shown you for the TMT data. And the only difference is that in this ion chromatogram figure, you can see the XIC peaks for light, medium, and heavy peptides, since the quantification is performed at MS1 level for select type of data. So to summary, today I have shown you the common practice for identifications of PTMs with PEAKS database search. And secondly, how to use PEAKS PTM function to discover unspecified and hidden modifications. And also the quantitative analysis of PTMs across different samples by using PEAKS PTM profiling function. And also I have showed you some new features related to PTM analysis, which will be implemented in the next version of PEAKS. And we hope that the information we provide you today can help you better perform your PTM analysis by using PEAKS software. And thank you very much for your attention.